Hello everyone, I am Shubhangi Kesarwani and today we are going to explore a very exciting topic from biology, that is the enzymes, which is also called as the biological catalyst. So now before we start, tell me one thing, when you eat your food, does it get digested immediately? No, right? So that is happening because our body has some helpers that make chemical reaction happens very fast. So these are called as enzymes. So by the end of this class, I think you will understand how these enzymes make our life possible and how they work and why we can't live without them, right? So what are enzymes? Enzymes are the biological catalyst, means the substance made by living organism that actually speeds up the chemical reactions without being used up by themselves, right? So most of the enzymes are proteins, and what they do, they actually help every reaction in our body, from digestion to respiration and to cell repair. Okay, for example, I'll tell you, um, the enzyme salivary amylase, okay, in our mouth breaks starch into sugar. Okay, that's why if you chew food like rice or bread for longer time, what happens? It actually starts tasting sweet. This happens, right? So what would happen if enzymes stop working in our body? Hmm? Then all the chemical reaction would become very slow. Okay, like digestion, breathing, or even heartbeat would stop. Life wouldn't be possible without it. Okay, so moving forward, these are the index we are going to study. Okay, and I have just told you what are enzymes, right? And let's see how enzyme help us in our daily life. Firstly, in digestion, enzymes like amylase, protease, and lipase break down food into the simple form. And in respiration, enzymes in mitochondria releases energy. During the DNA replication, enzymes like DNA polymerase and enzymes like DNA polymerase actually copy our genetic material. And in industries, as you can see here, in industries, enzymes are used in making the cheese, detergent, and even medicinal things. Okay, so enzymes are not just inside our body, right? So they are the part of many products we use. Okay, so have you seen a detergent pack when where it says like bioactive enzymes? Do you know what does it mean? So if you know the answer, that's very good, and if you don't, I'll tell you. Okay, it means that those washing powder contain enzymes that break down protein or fat stains. What will happen? Making the clothes cleaner. Okay, and what are the characteristics of enzymes? Okay, since we are studying about enzyme, so why it makes the enzyme so special? Because they are highly specific. What it means, each enzyme works only on a particular substrate. Okay, I'll tell you what are substrate and all. Okay, work and work under mild conditions, means normal body temperature and pH. It will only work when it is of normal temperature and the pH. In other characteristics of enzyme, that it is reusable, they don't get used up. Okay, and the fourth one is they are extremely fast. Okay, they can speed up the reaction by millions of times, which I have already mentioned, right? And so enzymes are like tiny biological machines working non-stop inside us. Okay, so if enzyme can be reused, okay, what does that mean to our body? So it means, I'll tell you, it means our body doesn't have to keep making the new enzymes every time or after every reaction. That means one enzyme can work again and again. That is the meaning of the reusing thing. Okay. And this is the question I have already mentioned. Okay. And these are the key features of enzymes, or you can say the characteristics I have already mentioned, as you can see in this slide. And this is the structure of enzyme. Okay. So every enzyme, every enzyme has a special region. Okay. Every enzyme has a special region which is called as active site. Okay. This is like a pocket or groove, okay, where the substrate, the molecule it acts upon fits perfectly. See, it is like lock and key. And the shape of this active site is unique for every enzyme. Why the shape of the active site important? Can you see here? So the answer is because only the right substrate can fit into it. If the shape changes, the enzyme won't work. It's like the lock and key for a particular thing. This is the substrate and this is the active site of the enzyme. And the shape is complementary to the substrate. Moving forward, so enzymes act like factories that keep working repeatedly. So now see how enzyme actually works. Step 1 is the substrate binds to the enzyme's active site. See, binding to the enzyme active site. And the second step is the enzyme convert the substrate into the product. So this is the final product which is getting formed. And the third step is the product is released and the enzymes remain unchanged. Okay. So enzyme acts like the factories that keep the working repeatedly. Okay. Does the enzyme get changed after the reaction? So the answer is no. Okay. So the enzyme stays the same every time and can be reused again, as we already mentioned. Okay. Moving forward, these are the examples like amylase, protease, lipase, and catalases. And these amylase, as you can see, amylase, what they do, it actually breaks starch into sugar. Okay. And protease breaks protein into amino acid. Lipase, what does lipase do? It breaks fat into fatty acid and glycerol. And water catalase breaks hydrogen peroxide into water and oxygen. Okay. So catalase is one of the it is not mentioned here in the slide, but I'll tell you that catalase is one of the fastest enzyme. Okay. It can break million of the molecule in seconds. Okay. So where the catalase found in our body? This is the question. So if you know the answer, that's very good. If you don't, I'll tell you. Okay. So it is found in the liver. It protects our cells from harmful hydrogen peroxide. Okay. So it protects our cells from harmful peroxide. Sorry. Okay. So this is the thing about the catalase. Okay. See, H2O2. This I have mentioned. Okay. Let's move forward. The types of enzyme. Okay. So based on their function, enzyme are classified into 6 types. Okay. Like oxidoreductases for oxidation and reduction reaction. Transferases transfer group between the molecules, and hydrolases break bonds using water like digestive enzyme. Lysases, what they do, they actually add or remove group without using the water. Okay, and isomerases, what they do, rearrange the atom within a molecule. And ligases joins two molecule using the ATP energy. Remember one thing, most enzyme name end with ase. Okay, in the last slide you have seen most of the even 90% of the enzyme ends with the word ase. Okay, which type helps in the digestion, can you tell me? So the answer is hydrolases, because they break food molecule using water. Okay, as you can see here, right? Let's move forward. So this is the lock and key hypothesis, which I have already discussed about the lock and key few slides back. So this idea was given by Emil Fischer. Okay, what he said, that the enzyme and substrate fits perfectly like lock and key. Okay, and this active site has a fixed shape. Okay, and only the correct substrate fits into it. If this rectangle piece is somewhat in the circle form, will it be able to fit in this area? The answer is no, it won't be able to fit. Okay, so the question is, if the substrate change the shape slightly, don't ask about the circle thing, a little bit the shape is different, will it able be to enzyme still the enzyme will still work? The answer is no. Just like a wrong key won't open a lock. Okay, the enzyme won't work if the substrate doesn't fit. Simple. Let's move forward. So another thing is induced fit model. So later Daniel Koshland explained a more thing about the flexible model, which is called as induced fit model. What does it mean? So he said that enzymes are not rigid. They slightly change shape when the substrate binds to fit even more better, like handshake that adjust for comfort, right? So what's the main difference between lock and key uh, and induced fit? In lock and key, what happens? In lock and key, the enzyme's shape is fixed. But in induced fit, in this model, what is happening? The enzyme adjusts its shape around the substrate. It can change a little bit because it is flexible. And what are the factors? Now let's understand what are the factors which are going to affect the enzyme performance. First one is temperature. Okay, first one is temperature. So every enzyme works best at the optimum temperature around 37 degrees Celsius in humans. All right. So if it is too low, enzyme slows down. So if it's too high, what will happen? It will get denatured and it will lose their shape and they will ultimately stop working. So what happened to enzyme activity when we get fever? What will happen? Do you have the answer? So if no, I'll tell you. Okay, so the answer is it slows down because the body temperature goes above optimum. Okay, it it basically works at the normal body temperature of the humans, like 37 degrees Celsius. Okay, now next thing which is affecting the enzyme activity is this pH. Okay, for each enzyme has its own optimum pH, right? For example, like in our stomach, that is pepsin, it actually works best when it is in acidic form. How much pH? That is 2, which is very acidic. Okay, while amylase, okay, works nearly neutral pH, around 7. Neutral pH is 7. Okay, so question is, if what if the pH becomes too high or too low? What will happen? The enzyme loses its activity, right? Its shape gets disrupted. Okay, and the third one is, the third one that is actually affecting the enzyme activity is the substrate concentration. Okay, at first more substrate means faster reaction, but after all the active sites are occupied, what happens? The rate of the level is little off. The enzymes become saturated. Okay, so this is the thing happening. Okay, then next one is the enzyme concentration, very important. Okay, more enzyme means more faster reaction, but only if enough substrate is available, otherwise it will not work. Okay, so these are all the things which are actually affecting the enzymatic activity. Okay, and few of them we have understood. Hmm? Next one is the enzyme inhibition. Okay, sometimes what happens is enzymes are blocked by certain chemicals. This is called as inhibition. Okay, so first one is competitive inhibition. Okay, the inhibitor and the substrate compete for the same active site. Okay, same active site, like two people trying to sit on one chair. Okay, example it is mentioned, like methotrexate inhibits the enzyme involved in the DNA synthesis. Okay, methotrexate inhibits an enzyme involved in the DNA synthesis. Okay, then next one is the non-competitive inhibition. What happens here? So here the inhibitor binds elsewhere, changing the enzyme shape so that the substrate can't fix. Can you see? It won't be able to fit. Okay, example like cyanide, okay, inhibits the respiratory enzymes, so that is why it is so toxic. Okay, we already know this cyanide. Okay, so so if we talk about, so what is the difference between the two types? Okay, this competitive and non-competitive one. So if I just give you the crisp explanation, what happens in competitive inhibition, the inhibitor competes on the same site. Okay, but in the non-competitive one, it binds somewhere else. See, it is binding somewhere else and change the shape. Okay, moving forward, enzymes are useful not only in our body but also around us as well. Firstly, in medicines. Okay, so used in the blood sugar test, like glucose oxidase, used as digestive enzyme supplement. Okay, then next one is what? In industries as well, renin renin in cheese making, zymase in brewing, and protease and lipase in detergents, used in the biodegradation and the environmental cleaning as well. Okay, so there is one question for you. So can you name any product at your home that uses enzymes? Okay, so the answer is yes. Biodetergents like we use, like Surf Excel or Ariel, they contain enzyme to remove the stains. Okay, so these are the things which I have mentioned. You can see in this slide how it is using. See, zymase, renin, detergents like Surf Excel and Ariel, and helping in the cleaning of environment. Okay, so let's quickly revise what we have learned today. Okay, so enzymes are biological catalysts made up of protein. They are very specific, and they are reusable and affected by the temperature, pH, and concentration. They also follow lock and key. They also follow lock and key model, and also using the induced fit model. They are used in digestion, respiration, medicine, and industries and whatnot. Okay, so without enzyme, life chemical reaction would be very slow to sustain life. Okay, there is one fun fact I would like to add is one molecule of catalase can break millions of hydrogen peroxide molecule, which I have already mentioned if you remember, and every second. That's faster than any machine we have built. Okay, which enzyme? Catalase. Okay, so remember, enzyme are the invisible worker that makes the life possible. Every heartbeat, every breath, and every thought in your brain happen because of the enzyme are at work. Okay, so thank you so much for learning with me today. I hope you have understand why enzymes are truly called the biological catalyst of life. Okay, so thank you very much.